So we're going to start with the introduction to embryology, a journey from a single cell to a human being. What is embryology first? Embryology is the study of the development of an organism in the uterus. So uterus we're talking about is the womb. So the study of the development of an organism in the uterus is called embryology. First, let's understand the principle of biology before starting to discuss about embryology. So what is biology? The word bio means life and logo means study. So embryology is now the study of how new life begins. So embryology is the study of how the biology of the life begins. Now, what is embryology in a simple term? It is the science that studies the origin and development of an organism from a single fertilized egg to its birth. So embryology is the study of an organism from fertilization. The fertilization is when the sperm meets the egg in the fallopian tube and they result to a baby birth. That is what we call embryology. Now let's discuss the process of embryology. So the first thing that starts with embryology is the fusion of the male gametes with the female gamete. The male gamete is called the sperm, while the female gamete is called the oocyte, ovum or the oval. So when they fuse together, when the nucleus fuse together, they result to what? A zygote. But before we proceed, let me tell you a story. And the story is, for you to be existing on this earth, you have fight too many battles. So for you to exist, the mother that gave birth to you have about 2 million eggs, 2 million oocytes. Eggs are oocytes. 2 million oocytes inside her ovary. And your father secretes or ejaculates 200 to 500 millions of sperm. So for you to exist, you have fight with numerous of sperms and egg for you to exist. So when these sperms and egg fuse together, we now form zygote. Now when this a zygote is a single cell that results from fertilization. So when these zygotes now start dividing and they start moving. And when they start dividing from a single cell to double cell to 4, 8, 16, we now form what we call a morula. A morula is a mitotic cell division of the zygote to form 16 cells are called a morula. So the process by which these cells are constantly divided immediately after fertilization to form zygote is what we call cleavage. Now, this cell has cytoplasms. Inside the cytoplasms, they are made up of fluid, and this fluid is what is responsible for feeding these eggs, these oocytes, this fertilized ovum. So a time will come where this cytoplasms or the nutrients is depleted. It can no more feed the egg, the fertilized eggs. What will happen? The egg will now start implementation. Implementation means when the morula starts cleaving or starts fusing itself to the uterus, where the cleavage will now fuse itself to the uterus. That is implementation. Now what happens? The fluid from the uterus, from the uterine cavity or from the endometrium will start entering inside the morula. So the process by which this fluid starts entering into the morula, it is now called a blastocyte. So a blastocyte is a result from fluid. When the fluid starts moving from the uterus to the morula, that's called blastocyte. So this blastocyte now differentiates. This is just an overview. These blastocytes now differentiate to form embryoblasts and trophoblasts. So the embryoblasts are the inner cell mass. Now make a deep observation of this embryoblast will now form the embryo, while the trophoblast will now form the placenta. Now, the embryoblast are the inner cell mass. So there are cells that are inside. So those cells that are inside forms the embryoblast, which will now later form the embryo. While those ones that are outside will now form trophoblast, which will now later form the placenta. So blastocytes are formed when fluid from the uterine endometrium, so when I say uterine endometrium, that is the uterus, starts moving towards the morula. So that process, blastocyte is formed, and each cell inside these blastocytes are called blastomeres. In biology is the science of development of an individual before birth. So I've given you so many divisions of this in biology. So embryology is the science of the development of an individual before birth is called what in biology, or the science that gives you the development of single fertilized egg to birth is called embryology. So let's start. We have lenses of embryology. So the least lenses of embryology means the subdivision of embryology. So the subdivision of embryology. So we have general embryology. So what are general embryology? General embryology is the process of early development from fertilization to the formation of germ layers. So study general embryology studies the processes of early development from fertilization to the formation of germ layers. So the germ layers are the ectoderm, the mesoderm, and the endoderm. Germ layers are the ectoderm, the mesoderm, and the endoderm. So what general embryology deals with the development from fertilization to the formation of what? Germ layers. Now the second is systematic embryology. So systematic embryology focuses on the development of the specific organ system. The development of the specific organ system is what we call systematic embryology. We are studying it, we are viewing it in terms of system, the digestive system, the cardiovascular system, the nervous system. So system by system is what we call systematic embryology. So we have comparative embryology. I know something is in your mind now. We are comparing and constructing the different stages of the development of an embryo in different species. So next we have experimental embryology. Experimental embryology uses experimental. We are using experiments to understand the mechanism of development like cell signaling. We are trying to understand embryology. This is more of practical embryology. We are using experiments to understand the embryology study of individual inside the uterus. So we have teratology. The teratology is the study of the congenital anomalies and the factors that causes them. Teratology is like the abnormalities of embryology. What causes the abnormalities of a child not developing well, of an organism not developing well? What causes it? You see a child without well developed respiratory system, you see a child with so many abnormalities when a child is not developing normally. So this teratology deals with that aspect, the study of embryology, and what causes the abnormalities of that embryology. So we have the biochemical and molecular embryology. It investigates the genetic and molecular events that control development. So we have the clinical significance. Why embryology is cornerstone of medicine. So why are we studying embryology? We are studying embryology. One is to understand congenital anomalies. Most major birth defects occurs during third to 8 weeks. So Knowledge of normal development is key to understanding abnormal development. So for you to understand the abnormalities of embryology, you must have understand the normal way of by which embryology is formed. For you to understand pathology, what causes disease in human, you must have read histology. You must have understand the way the normal cells and tissue is before going to the abnormalities of the tissues and cells. So before you go to the congenital effects and abnormalities, you must have understand the way it works. And during 3 to 8 weeks, what usually happens is development of organ. Organogenesis takes place. So organogenesis means development of organ. So during this period, what happens is what? Development of organ. Now, another thing is the obstetrics and the gynecology. These are doctors or professionals that inclined or help or they are essential for managing pregnancy. They are the one that take cares of pregnant woman, understanding ectopic pregnancies and using techniques like in vitro fertilization. You have pediatrics. So what are pediatrics? These are medicals professionals that take care of children, child, newborns. They take care of child or children's. So you have the surgery or radiology, understanding the anatomical variations and origins of structural defects is crucial for surgical planning and interpreting medical images. Now, can you look at this woman here? This woman is pregnant, so we are using radiology to radiological technical imaging to determine whether the baby is developing well or not. So we are trying to check whether the baby is developing well or not. And note that when a baby or a child or an embryo or a developing child inside the uterus is exposed to radiations, it leads to abnormalities in the baby. It leads to congenital defect in the baby. It leads to when the baby, it leads to so many disadvantages of the baby. It might lead to the abnormality of organogenesis. So you should be careful when a pregnant or a woman, when a woman is pregnant, she is not advised to go to where radiations are, or she's not exposed. She's not good to be exposed to what? Radiations, because it will affect the baby. So let's go over to the genesis, the gonads and the gametes. So this is where embryology begins. Reproduction, the biological process by which, so what is reproduction? Reproduction is the biological process by which new individuals are produced. Why? To ensure the continuity of life. We are producing new individual so that we can ensure the continuity of life. So that is why we are reproducing. That is why your mom and dad will keep on doing something, because they want to reproduce in order to ensure the continuity of life. So first, what are gonads and what are gametes? Gonads are the primary reproductive organs. Let's make it clear. Gonad is just area where gametes are produced. They are areas, paired areas where gametes are produced. So when you heard of gonad, they are just responsible of producing what? Gametes. Example, the male has testes, and this testis is where spermatozoa are produced or sperms are produced. The female have ovaries, and what is the ovaries doing? The ovaries are the region or area in the uterus where eggs are produced. So gonads are gamete producing cells. They are gamete producing cells. So we have been talking about gonads are gamete producing cells. Therefore, what are gametes? Gametes, they are specialized reproductive cells which are haploid. Gametes are called sex cell, so they are responsible for producing the individual. So gametes are what we use to produce individuals, and gametes are haploid. We have what we call haploid and we have what we call diploid. Diploid is when an individual or cell has a complete chromosome, and a complete chromosome is 46 chromosome, while gamete has haploid chromosome because they have undergo meiotic cell division. A meiotic cell division is a division of cell in which the daughter cells have half number of chromosome of the parent cells that produce them. So the gametes has 23 chromosomes, meaning the sperm has 23 chromosomes and the female, which is the oocyte or the egg, has 23 chromosomes. So when they come and fuse together, they will now form 46 chromosomes, making it what? A diploid zygote. So let's talk about gametogenesis. What is gametogenesis? Gametogenesis, the word gametes means gametes. Why? Genesis means we are producing, we are forming, we are synthesizing. So if you combine it, gametogenesis is a process of forming gametes through meiosis. So we are forming gametes through meiosis. We have what we call mitotic cell division and we have what we call meiotic cell division. Mitotic cell division, the daughter cell will have the same number of chromosomes as the parent cell that produces them, while in meiotic cell division, the daughter cell will have half number of chromosomes as the parent cell that produces them. So let's gametogenesis is divided into two, which are the spermatogenesis and the oogenesis. So what is spermatogenesis? Spermatogenesis is the process by which we produce sperm, and oogenesis is a process by which we produce oocytes and a polar body. So let's start. Let me just give you a quick review of this gametogenesis and oogenesis. So let's start with gametogenesis. Now, first, gametogenesis, which are formed from primordial germ cells, which will now produce spermatogonia. This spermatogonia will now come and produce, we undergo mitotic cell division. I hope when I'm talking about mitotic and meiotic cell division, you now know what they mean. Mitotic, they have the same, they are diploid and have the same number of chromosomes as the parents that produces them. While meiotic, they are haploid and have the number of chromosomes as the parents that produces them. So primary spermatocytes will undergo meiotic cell division to produce secondary spermatocytes. These secondary spermatocytes will now undergo meiotic cell division, the second meiotic cell division to produce sperm. This is just an overview, it's not the complete, and we are just going through, we are just introducing you. So with time, we start understanding all these things better. So the primary spermatocytes also do the same thing. They undergo meiotic cell division to produce secondary oocytes, and they now undergo another meiotic division to produce what? Functional oocytes, which is only one. So we have the human timeline, the mapping our development. We are mapping our development. So development, what is development? The development is the advancement of an individual. So when an individual starts advancing, starts growing and starts advancing, we say this is what? It is development. When it starts forming complex structure, we say this is development. So the entire sequence of events in a life of an individual from fertilization to death is what we call development. So from the point of fertilization to the point of death or old stage is what we call development. And also have what we call stage of human life. So the development is divided into two major periods, which are the prenatal period and the postnatal period. Two major periods, which are the prenatal period and the postnatal period. Now, what happens during the prenatal period? and what happens during the postnatal period. First question is, what is prenatal period and what is postnatal period? But before I explain that, let us explain what is natal. Natal means birth. So when we talk about natal means birth. So when we talk about prenatal period means before birth. What is happening before birth? And postnatal period means after birth. What is happening after birth? So the human development is divided into two, which is the prenatal period, which is before birth, and the postnatal period, which is after birth. So what happens during the prenatal period? As you can see, the first things that happen is what? Fertilization, in which the sperm fuses with the ovum or the oocytes. They now result to what? Zygote. The zygote now undergo mitotic cell division. With that process of rapid mitotic cell division is what we call cleavage, and during this cleavage they now form a morula. Now the morula, what does now happen? Fluid starts entering into the morula from the uterine endometrium. This now form the embryo. The embryo now further undergo to form the fertile period, which is the fetus. The zygote results to fetus. The fetus, when we talk about fetus, fetus is from the ninth week of fertilization to the 9 months, or about the 37 weeks. So this is what happens during the prenatal period. So what happens during the postnatal period is we form a neonate. From a neonate we form, like, which is babies, child. From child we form adult. From adult, elderly person. From elderly person, old. Then death now occur. So these are the major things that happen in the prenatal period. The period of development before birth, it encompasses fertilization, meaning it contains fertilization, embryonic development, and fetal growth. The focus is on formation. So what happens during the prenatal period is fertilization, which we all know. The first thing that occur is fertilization. Second is the embryonic development. So when we talk about embryonic development, is from the third week to the eighth week is embryonic. And what is happening? Mostly is what? Organogenesis. Organogenesis means formation of organ, or formation of what? Germ layers. So what does now happen? We now resort to fertile growth. Fertile growth is after ninth week of fertilization to 37th week is what's called the fertile growth. The fetus starts developing. So at that period, from third week to eighth week, we call it an embryo. That's what we call it embryonic development. While from ninth week to 37th week, we now call it fertile, or the fetal, or the fetus, which we now refer to as the fetal growth. So what happens during the postnatal period? The period of development after birth is what's called postnatal period. And what's happening? It includes infancy, childhood, adolescence, adulthood, and old age. They focus on growth and aging. Childhood, adolescence, adulthood, and old age focus on the growth and what? Aging. So what you just have to have in mind is, during the postnatal period, all these things takes place. Now, what next is the prenatal period? So what happens in the prenatal period? The prenatal period is the first 8 weeks we are building the foundation. What happens is, three things happens. One is the pre-implantation, or we also call it what? The pre-embryonic period. The second is what? The embryonic period. And the third is the fetal period. So what happens during the pre-implantation? What you have to know is, we call it the pre-embryonic. Pre means before embryo, and pre-implantation means before implantation. Before we fuses it with the uterus. Implantation means when these blastocytes have fused with the uterus, have entered into the endometrium of the uterus. So before this occur, pre-implantation has to occur. So what happens during the pre-implantation is a rapid sequence of critical events before the embryo attached to the uterus. At the pre-implementation, meaning before we form the embryo, what happens is we start attaching the embryo to the uterus. We call it embryo from the third week to the eighth week. So what happens is the embryo starts attaching itself to the uterus. And during this process, what happens? Fertilization. After fertilization, what again happen? Cleavage. What is cleavage? The mitotic and rapid cell division that takes place immediately after the formation of zygote, which will now result to the formation of what? Morula. Now, when fluid starts entering from the endometrium to the morula, we now call it a blastocyte, and cells of these blastocytes are called blastomeres. Now, what happens? Implantation begins. Now, take a look at this diagram. We have the inner cell mass. This inner cell mass, so the inner cell mass remain just the inner cell. So this is a diagram of a blastocyte. So blastocytes have just differentiated into embryoblast and the trophoblast. Now this is the trophoblast, and this is called the blastocyte cavity. It's called the blastocyte cavity because cavity means space within. Just a space is what's called cavity. So that space, what's filled there is the fluid that have entered from the uterine to the morula to now form a blastocyte. And due to this reason, the cells start differentiating. Due to this reason, the cells start differentiating. And what will the cell differentiate? The cell will form inner mass. Inner mass means those cells that are inside, while outer mass means those cells that are at outermost, those cells that are exposed to the uterine. So the inner one will now form the embryoblast. So what will now happen to the embryoblast? The embryoblast will now further go and form the embryo. So the outer one will now form the trophoblast. What's happened to the trophoblast? The trophoblast will now further go and form the placenta. Now take a look at this diagram. This is the uterine cavity. This is the uterus. So what is happening? This is where implantation is about to occur. And those cells that are inside are called the embryoblast, while those ones that are outside or that are exposed or that are attached to the uterine cavity or the endometrium or the uterine endometrium is what we call the trophoblast, which will now form the placenta. Don't forget that. But that is what you just have to know. I'm just opening your idea. So the second thing that happens in the prenatal period is the embryonic period, which takes place from the 2 to 8 weeks. So what happens in the embryonic period? The embryonic period means this is the formation of the embryo. So the most critical period of development where all major organs system begins to form, organogenesis. So when your organs begins to form, when your brain, your anything organ, your liver, your stomach, everything, when they start the beginning of the formation of these organs is what we call organogenesis. And which period is that? The embryonic period. And this is where abnormalities can easily occur, and this is where you are carefully checked, because any mistake can lead to formation of, or can lead to production that look like another thing. So gastrulation, what are the main key events? Gastrulation and organogenesis. And what is gastrulation? Formation of three primordial germ layers. Just have it in mind, gastrulation occurs, and during gastrulation we are forming the germ layers. And what are the germ layers? The ectoderm, the mesoderm, and the endoderm. That's what's happening. And what again is happening? Organogenesis, the differentiation of germ layers into specific organs and tissue. The germ layers start differentiating into embryo and placenta, and the germ layer starts forming what? Organs. The embryo is extremely sensitive to teratogens during this time. So those teratogens are like what causes abnormalities to the embryo. So the third thing that happen is now the fetal period. Remember, three things happen during the prenatal period. The first, the pre-implementation. Second is the embryonic period, and third is the fetal period. And the fetal period occurs from 9th week to 37th week. So that is the fetus. So after 8 week, which is after 8 week, the embryo have been formed, organogenesis have taken place. The developing woman is called a fetus. So after 8 weeks, from the ninth week, we now call it a fetus. Due to this reason, we now call it what? The fertile period. The focus shifting, the focus shifts from forming organs to growing and maturing them. So during the embryonic period, what happens? We start forming organ. So after the 8 week, the ninth week, we are done, we are through with the formation of the organ. What next now happen? We start developing them. The advancement of the organs is what we call development. When we start advancing them to complex structures, that's what we call development. So organogenesis have finished, so we have started developing them. So what are the key characteristics that occur during this fertile period? Is rapid growth, significant increase in size, length, weight, especially in the last trimesters. Remember, the fertile period is divided into three: the first trimester, the second trimester, and the third trimester. They are just they are just period that is trying to explain the gestation period. The gestation is the pregnancy period. So when a woman is pregnant, the trimesters are divided into three, and these three are trying to explain what is happening during the gestation period, also known as the pregnancy period. What next happen? Maturation of tissues and organ. Remember, at the fertile period, we are done with the formation of organ. What we are now doing is to develop them, to advance them. So tissue and organs system established in embryonic period becomes functional. What are the system established in embryonic period? The embryoblast and the trophoblast. So what are they going to do? The embryoblast will now form the embryo, and the trophoblast will now form the placenta. Changes in proportion. The head, which was initially very large relative to the body, becomes more proportional over time. So the head starts having the same shape, like starts they become proportional to each other with the body. So less vulnerable to major structure defects, but still susceptible to issues affecting growth and functional development. Example is the brain. So when we are exposed to teratogens, example are radiations, dey affect dis embryo during the embryonic period. But once we cross the embryonic period, dey still affect dem during the fertile period, but the way by which dey affect dem are reduced. The way by which dey affect dem are reduced compared to the embryonic period. So as you can see, what's happening? Changes in proportion, changes in size, dey start developing dem more. Their body, their head, everything starts matching during the fertile period. So let's talk about what happens during the postnatal period. We have talked about what happens before birth, which is the prenatal period. So what happens before the prenatal period are the pre-implantation, also known as the pre-embryonic period, the embryonic period, and the fetal period. Now, what happens during the postnatal period, also known as the after birth? What happens? Newborn, formation of neonates. What are neonates? After birth, from 0 to 28 days, we call them neonates. When a child is giving birth, from 0 to 28 days, we call them neonates, newborns, neonates. And these are the processes that occur during the postnatal period. So what else now occur? The baby keep on developing. So 1 month to 1 year, we now call them infants. So the study of this region is now called what? Infant. The period is now called the infant. From 1 month to 1 year, we call them infants. From 0 days, or from the time they are giving birth to 28 days, we call them neonates. From 1 month to 1 year, we call them infants. From 1 year to 12 or 13 years, we call them childhood. That is child. And doctors that take child are called pediatrics. They take care of children. So puberty takes place from 17 to 20 years. Puberty takes place from 17 to 20 years. While adolescence takes place from 17 to 20 years. While puberty takes place from 12 to 16 years. Puberty takes place from 12 to 16 years. Adolescence takes place from 17 to 20 years. Then adult is from 21 to 40 years. So you are called an adult when you reach from, or when your age ranges from 21 to 40 years. While middle age is from 40 to 60 years, and old age is from 60 years to what? Death. So let's recap. Neonates 0 to 28 days, infants 1 month to 1 year, childhood 1 year to what? 12 or 13 years, puberty from 12 years to 16 years, adolescence from 17 to 20 years, and this is where spermatogenesis occur. Some also start from 16, some 15, but women start from from the fertile period. Oogenesis have already started taking place. Remember, oogenesis is the formation of gametes. So women, before they are giving birth, oogenesis have already started taking place. While male, oogenesis begins at around the puberty, around 14, around the puberty to adolescence age. So middle age is from 40 to 60, and old age is from what? 60 years. So let's talk about the individual and species ontogeny and phylogeny. Remember, we are still in the introduction to embryology. So the question is, what is ontogeny and what is phylogeny? Ontogeny simply means the study of the complete life cycle of an individual. So when you study the life cycle from fertilization to zygote, from zygote to morula, from morula to blastocyte, from blastocyte to embryo, from embryo to fetal, to fetus, from fetus to a child, from child, or from fetus to neonates, from neonates to infants, from infants to children, from children to puberty, from puberty to adolescence, from adolescence to middle age, from middle age, or from adult to middle age, from middle age to old age, from old age to death is also called ontogeny. The complete life cycle of an individual is also called ontogeny. So phylogeny is the phylogeny is the evolutionary history and development of species or group of organisms. So when you study the evolutionary history of a species or group of organisms, we call that phylogeny. So think of it like that: the developmental story of humanity, and the ontogeny, your personal development story. So the connection: ontogeny recapitulates phylogeny, is a historical theory suggesting an organism development reflects its species evolutionary stages. While not strictly true, developmental similarities across species do reveal evolutionary relationships. Now, let's talk about the clinical focus, which is the prematurity or and the viability. So let's talk about the prematurity. What are prematurity? Or prematurity, we are just trying to say the premature delivery. Premature delivery. So delivery that occurs before the start of 37 weeks of pregnancy is called premature delivery. So have you heard of preterm? Preterm are child or embryo or fetus that have not reached 37 weeks. So when they are giving birth, or when they are out of the uterus before 37 weeks, we call them preterms or premature delivery. Is when they are out before the 37 weeks of pregnancy, we call them preterm. So abortion also form under these preterms. When a child is giving birth or is out of the uterus before the 37 week, either 36 week or 36 and something, we all call them preterms because they are out of the uterus before 37 weeks. So how can we prevent this? First, what causes this is alcohols. So a woman that is smoking, a woman that consume alcohols causes what? These preterms So when you see a woman that is pregnant, please kindly advise her to stay away from alcohol, and if she's a type that smoke, let her stop smoking. The type that smoke, let her stop smoking, because they causes what we call preterms. So we have viability of fetus. So what are viability of fetus? Viability of fetus is the ability of fetus to survive outside the uterus. So when a fetus or a child or an embryo or a fetus survive outside the uterus, or the ability of them to survive outside the uterus before 37 weeks, is what we call the viability of fetus. And they can only survive depending, or this depends heavily on lungs and the central nervous system maturation. Approximately approximate survival chances: when these fetus are out of the uterus before 24 weeks, they have 40 to 50% chance of surviving for those that is weighing 500 grams or more. But when these fetus have stayed up to 28 weeks, they have 90% chance of surviving for those that have weighs more than 800 grams. So in conclusion, viability of fetus is the ability of fetus to survive outside the uterus. Let's talk about the multiple pregnancy. Have you heard of twins, identical twins and fraternal twins? I'm going to make you understand what is the difference between identical and fraternal twins. So, definition: the simultaneous development of two or more fetus in the uterus. Now, what comes into your mind when you talk about identical twins? Identical twins is when sperm meets the ovum and fertilize the ovum. Now, this ovum now form a zygote. Now, when this zygote completely divides into two, we call it identical twin. Remember, when zygote is formed, they undergo mitotic cell division to start producing morula by a process known as cleavage. But in identical twins, these zygotes will divide completely into two equal parts, into two. As they divide into two, they now form two different something, two different zygote, fertilized zygote. They start developing, and as they start developing, this is what's called identical twins, or we call it monozygotic twins. Monozygotic twins is also called identical twins because they are mono. Only one. Mono means one. Only one sperm and one egg fertilize. Only one sperm fertilize the egg, and only one egg is involved. But in fraternal or dizygotic twins, one sperm fertilize two sperms fertilize two egg, whereby a sperm will come and fertilize an egg, and another sperm will also come and fertilize an egg. So in fraternal twins, that's why in fraternal twins they do not look alike. They are so different. You will not even be able to recognize whether they are twins unless they say something like that. So in fraternal twins, two different sperm come and fertilize two different egg. While in identical twins or monozygotic twins, only one sperm and one egg is involved, and they fuse together and fertilize to form a zygote. And when they form a zygote, they now divide into two to now form two different zygotes that will now start developing in the uterine endometrium. So we have what we call differentiation and organizers. So what are differentiations and what are organizers? Let's start with differentiations. Differentiation: first, which cells undergo differentiation? Stem cells undergoes differentiations. Stem cells undergoes differentiations. And differentiation is a process by which unspecialized embryonic cells acquire features of specialized cells, choosing their carriers to become muscle, nerve, skin, etc. So just what you have to know in mind or have in mind is differentiation is the process by which unspecialized cells, they do not have a specific function, differentiate. Differentiate means they undergo mitotic cell division or rapid cell division to form more of themselves. It's either they're forming more of themselves or they are forming a specialized cell. This specialized cell will now perform a specific function. Example, stem cells differentiate in order to form other sperm cell, or they now form a special cell known as the neurons, the muscles, and the blood cells, which will now perform a specific function. The neurons perform a specific function by transmitting signals. Muscles produce movement, and blood cells also fight against unwanted organisms in the body. So we have types of differentiation. We have hemodifferentiation. Formation of the blood cells are called hemodifferentiation. Histodifferentiation, formation of specialized tissue. Hemo, blood. Histo, have you heard of histology? Study of the cell or the tissue of a body. So that is how we derive that word. Histodifferentiation, formation of specialized tissue. Organodifferentiation, you have heard of the word organs, formation of organs, formation of organ from stem cell. Functional differentiation, the point at which cells or organs become functionally mature. So we have organizers. Organizer, a group of cells in embryo that act as master architects, including and patterning the development of neighboring tissues. So the function of these organizers in the embryo is to produce cells that will direct the and arrange the developing tissues. So they produce cells that will direct and arrange the developing tissues. That's what's called organize. Organize just direct and arrange the developing tissues in the embryos. How? Example, they are using embryos to form organs. That process is called organogenesis. So what the organizers are doing, they are using cells to control the formation of this organ, which we call organogenesis. You have classic example. The primitive node can induce a second body axis, signaling the molecules. So the miracle cells, a closer look at the stem cells. So the stem cell are cells that can form all types of cells, including the embryo and the extraembryonic tissues. Remember, the embryo forms the embryo, and the extraembryonic tissues include the placenta and the rest, and the amniotic cavity. Just know that the extraembryonic tissues includes the placenta. So totipotent stem cells are cells that can form embryo and extraembryonic tissue. Example, cells from the embryo up to its cell stage. So cells from the embryo can form this. So you have pluripotent. These are cells that can form all types of cells except from the extraembryonic tissues. So they can form all the embryonic tissues, but they cannot form the extraembryonic tissue. So for this reason, what is the difference between totipotent and pluripotent? Totipotent can form embryonic and extraembryonic tissue, while pluripotent can only form the embryonic tissue and cannot form extraembryonic tissue. Embryonic stem cells from the examples are the embryonic stem cells from the inner cell mass of the blastocytes. Remember, totipotent example, we have the blastocytes can form the trophoblast, which will now form the placenta, and the embryoblast, which will now form the embryo. While pluripotent example of the stem cell is the embryo, which is the inner cell mass of the blastocytes. They can only form embryo, they cannot form the placenta. So they can only form the embryonic tissue, they cannot form outer embryonic tissues. As you see in totipotent, they can form trophoblast, and trophoblast form extra embryonic tissues, which are the what? The placenta. So you have the multipotent can form a limited range of different cell types within specific tissues or organ. So they are limited. Adult stems such as hematopoietic stem cells. So what are the clinical importance of these stem cells, or of looking of a closer look of these stem cells? Stem cell therapy holds promise for repairing damaged tissues and treating disease like Parkinson, diabetes, and spinal cord injuries. Stem cell therapy hold promise. So stem cell therapy, what are they doing? Their work is to repair damaged tissue, treating disease like Parkinson, diabetes, and spinal cord injuries. So we have the summary. So let's look at the summary. The summary is for first is formation of zygotes from fertilization. Second is blastocyte, then implementation that takes place, gastrulation that takes place, growth and maturation, and death that takes place. So what happens at the beginning? The embryo traces the journey from the fusion of the two gametes, sperm and oocyte, to form zygote. So first is what? Fertilization occur. Second, which is the first week, the zygote undergoes cleavage to form a morula, then a blastocyte. A blastocyte is formed when fluid starts entering into the morula, which implants in the uterus. Now, from there, in the clinical corners, understanding this developmental timeline is essential for diagnosing congenital anomalies. Anomalies there means abnormalities, and for clinical practice in obstetrics, pediatrics, and surgery. So what next now happen? The critical period. The embryonic period is defined by gastrulation. The embryonic period is what? What happens? Gastrulation. And during gastrulation, three things happens. Formation of the germ layers, which are the ectoderm, mesoderm, and the endoderm. And thirdly is what? Organogenesis. What happens? Like what you have to just know is during the critical period, which is the embryonic period, what happens is gastrulation and organogenesis. And gastrulation means formation of germ layers, which are the ectoderm, the mesoderm, and the endoderm. And organogenesis, which are the formation of what? Organs. Growth and maturation. So the organs now develop and become more advanced. That's what's called by growth and maturation. Next thing is now what? The concepts like stem cells and molecular signal are at the front of modern medicines. So this is just the advancement of medicines. So let's test your knowledge. What is the term for the study of the causes of birth defects? So here you have to think. So what is the term for the study of the causes of the birth defects is what? So what causes birth defects? Radiations causes birth defects. And examples of these radiations, as I've said, is teratogens is what called radiations. So the study of the causes of birth defects is called teratology. Teratology, because radiations are examples of teratogens, and the study of this is now called teratology. During which developmental period are all major organ system. So during which developmental period are all major organ systems established, making the embryo most susceptible to teratogens. So the question is just asking you is, which period are organs? Where begins the formation of organs? You don't have to think of that, which is what? Embryonic period. What happens in the embryonic period? Formation of organ, which takes from how many weeks? From third to what? A week. So we now have the fraternal or dizygotic, the fraternal or dizygotic twins originates from. So where does it originate from? Is it from a sperm which fused with an egg, or when two different sperm comes and fuse with two different egg? So when two different sperm fuses with two different egg, also known as what? Oocytes, we can call it what? Fraternal twin. So a stem cell can differentiate into any cell type of embryo proper, but not the placenta tissue is described as embryo. So a stem cell that can differentiate into any cell type of embryo proper, but the placenta tissue is but not the placenta tissue is described as what? Pluripotent. Remember, multipotent is when stem cell form both embryoblasts, both embryonic tissue and extraembryonic tissue, while pluripotent is when the stem can only form embryonic tissues. It cannot form extraembryonic tissues, which are the what? The placenta. So the embryonic tissues will now further develop to form the what? The placenta. So you see, they didn't come with extraembryonic tissue, they come with what? Placenta. That's why you should be careful. So we have the fifth question. What key event marks the transition from embryonic period to the fetal period? What key event marks it? The completion of what? Organogenesis. Because what happens in the fetal period is the advancement of organogenesis. So during this period, the completion of organogenesis have been completely what? Complete. So organogenesis have been completely complete. Therefore, what happens during embryonic period of the fetal period is the formation of organogenesis. Question: what are the two primary cell population of blastocytes and what do they become? So what are the two primary cells of blastocytes? Remember that I told you that blastocytes will further go and come and form inner cell mass, and this inner cell mass and outer cell mass. So these are the outer cell mass and these are the inner cell mass. So the outer cell mass will now form trophoblast and the inner cell mass will now form embryoblast. The embryoblast will now become the embryo while the trophoblast will now become the placenta. So the process of forming male and female gametes is called, this is a very simple question. Gametogenesis is the process by which you form male and female gametes. Question 8. A pregnancy that implants in the uterine tube instead of the uterus is called an ectopic pregnancy. How? Ectopic pregnancy is caused when the cilia becomes immobile. Immobile means the cilia can no more move. So the egg will not be able to move, the cilia will not be able to move the egg. Therefore, the sperm will now come and meet this egg at the ampulla, which is generally where fertilization takes place. But the egg cannot further move. So what will now happen? The egg will now start developing into the ampulla and cause the rupture of this ampulla, leading to bleeding and sometimes can lead to death. So an ectopic pregnancy results to when the cilia is not able to move and fertilization now takes place in the ampulla, and the development of these zygotes in the ampulla leads to ectopic pregnancy. Don't forget to like, subscribe and hit the notification bell for more videos like this on Sagacious Learners.